everyone, I'm going to go through the study guide for lecture exam 1. This is on chapter 20, 21, and 17, the lymphatic system, the immune system, and blood. So chapter 20 is on the lymphatic system, and there's two major objectives for this chapter. The first one is list the major components or structures of the lymphatic system and their functions. I've listed here the major components or structures, and their function is listed right in the PowerPoint script. So the structures are bone marrow, the spleen, the thymus, lymph nodes, tonsils, the appendix, and other lymphoid tissues that are found throughout the body, such as MALT, the mucosa-associated lymphatic tissue that is found throughout the mucous membranes, such as the gastrointestinal tract and the respiratory system. So you can find these functions going pretty much in order at the beginning of the PowerPoint script for chapter 20. I put a little note here that the thymus is actually quite large prenatally and in infants and children because remember, T cells mature here. So of course that's part of the lymphatic system. T lymphocytes, remember, are the T cells, and it shrinks as we age. It does produce some hormones which are actually classified as paracrines because they act locally. And these are involved in normal development of T lymphocytes. But we will be talking more about hormones when we get to the endocrine system. The second major objective for chapter 20 is to identify the source of lymph and the mechanisms for transporting it in the body. And the hint right here is actually the answer. Lymph, the fluid that travels throughout the lymphatic system, is produced via filtration of fluid at the blood capillary level. And the lymph primarily moves passively via the movement of the local skeletal muscles through what we call lymphatics. Lymphatics are the lymphatic vessels from the smallest, the lymphatic capillaries, up to the collecting vessels, and then lymphatic ducts, and then the largest lymphatic vessel is called a trunk, or the largest lymph passageway. The lymphatic trunks are the major trunks that will bring lymph back into the circulatory system. And also keep in mind that lacteals, which are discussed in the chapter, are small lymphatic capillaries that will absorb fats or lipids from the gastrointestinal tract. And we will come back to them when we get to that unit, the digestive system. Okay, the third objective, now we're on chapter 21, and this is really a discussion of the immunity, our protection against disease or against pathogens entering the body. Also against cancer cells. They have found out that we have a healthy immune system which will destroy cancer cells before they get a chance to multiply. So chapter 21 talks about immunity and the immune system, which is the job of all of the white blood cells. So when we get to chapter 17, we don't really talk much about white blood cells because there are many white blood cells involved in the immune system. So chapter 21 starts out with very straightforward immunity, which is actually provided by the barriers on the outside of the body, both the skin and the mucous membranes that are open to the outside of the body at orifices. And anyway, the barriers would include intact skin and mucous membrane and some of the chemicals that they secrete, such as the stomach acid, which is antibacterial. Both perspiration and saliva and tears in the eyes have some antibacterial properties to them. So the first line of defense, and this is considered innate because we're born with it. We're born with these barrier systems. It actually will keep the pathogen out of the body. So then, if any of the pathogens enter into or breach those barriers and get into the tissues, the second line of defense is called inflammation. And this is also innate and it's non-specific. So there are actually 4 signs of inflammation that are listed in your PowerPoint, and understand what these are caused by. Remember, redness, swelling, pain or itching, and warmth. And so I talked about that also in chapter 21 near the beginning. Now we have another objective. This is the 4th of the study guide, the 4th objective, the second one in chapter 21. And this is to describe the specific immune responses employed by the human body. This is our 3rd line of defense, and it's considered adaptive, meaning the body learns how to fight specifically against a specific antigen or a specific invader in the body. So we're not born with this, but we are born, of course, with the ability to form this adaptive immunity. And this is the job of your T cells or T lymphocytes and the B cells or B lymphocytes. B lymphocytes, these will engage in what we call humorally or antibody-mediated immunity, meaning that the B lymphocytes are going to fight the pathogen by creating antibodies, which will then target the antigens and mark them for destruction. So the B lymphocytes are one branch, and then we have, and remember, we also call these B cells. The cell-mediated immunity is engaged in by the T lymphocytes or T cells. And the bulk of the PowerPoint for chapter 21 actually discusses T lymphocytes in depth and also B lymphocytes in depth. The 5th objective for this study guide is define antigen and antibody. Remember, antibodies are made by B lymphocytes, and there is a discussion about what is an antibody. Remember, they are roughly Y-shaped. There's a stem and then the 2 arms that stick up that are relatively stable, and it's only the tips or the ends of the arms of the Y-shaped antibody that will be formed specifically to fit an antibody once that antibody is encountered in the body. There's actually 5 different categories of antibody. These are all called immune globulins, and there's several different classes that are discussed in your PowerPoint. So find that discussion and just know generally what those categories are, and that is the antibody. And remember, the antibodies are made by the B cells. And then I'd like you to understand or be able to briefly describe or recognize questions dealing with these terms right here. So the first terms are natural killer cells, NK cells they're called. And these act similar to a macrophage, which is a big giant cell. Macrophages, remember, will break down, literally eating the offending organism or toxin that's entered the body that's been identified as a foreign molecule, because there's an antigen that can be identified as foreign. Complement and interferons, and I listed these down here. These, remember, are the chemicals that enhance the immune system and lead to inflammation. One of the interferons actually will cause the body to develop a fever, an increased temperature by basically setting the thermostat higher. Our thermostat, in air quotes, would be located in the hypothalamus in the brain, and it will basically turn up the set point for our body temperature to be greater than 98.6 Fahrenheit or 37 degrees Celsius. And this fever actually it can be advantageous in that it is antibacterial. It inhibits bacterial growth or multiplication. And it also increases our metabolism and allows us to fight infection more effectively. Now we are up to the 2 types of B cells, or B lymphocytes. Plasma B cells, which are actually the cells that are responsible for producing and secreting antibody. Once the antigen has been identified in the body, an antibody specific to that antigen will be created by a B cell, and it will clone itself many times, and then those plasma B cells are the B cells that are actually going to produce and secrete the antibody. The memory B cells are, a memory B cell will be formed that actually keeps the memory or the record of that antigen that the body has encountered and how to make the antibody in order to neutralize that antigen. So the first time that you encounter a disease causing, like a pathogen, it takes a little while for the body to learn how to make the antibodies against that antigen. If it could take several days. In the meantime, if it's a very virulent pathogen, it may threaten the life of a person, or you could get really sick with it. But the next time, if you survive it, the next time that the body encounters that antigen, these memory B cells will have the record of how to immediately signal to the plasma B cells to make a lot of the antibody and secrete it and neutralize the antigen before it can get you sick. Now, those are the 2 kinds of B cells. Then over on the right here, I have the 4 types of T cells or T lymphocytes. The cytotoxic or killer T cells are actually equipped to do what's called a touch-kill mechanism. They release a chemical called perforin, which actually perforates the pathogen or the foreign cell. And so the T cells engage, remember, in this cell-mediated approach to getting rid of a foreign cell. And so the cells that actually engage in that are called the killer T cells or cytotoxic T cells. Then we have memory T cells, and just as we have memory B cells, memory T cells will keep a record of the memory of the encounter, and will basically have that record of what the foreign cell or the pathogen looks like, and how the body has developed a strategy in order to neutralize that cell or get rid of it before it actually can make you sick. Then the 3rd kind of T cell is called a regulatory or a suppressor T cell. These cells, remember, will signal to the body to stop fighting against a perceived enemy, because it can this could lead to what you might think of as friendly fire in a war, where the targets can become your own cells. Now, T cells, remember, as they develop, they need to learn how to recognize your own self cells, the markers on your own cells that tells the body, these are my cells, they belong here, don't destroy them. And so the regulatory or suppressor T cells are involved during an encounter with a pathogen or a foreign cell that tells the body, okay, the threat is over, so stop fighting this infection. Then we have the helper T cells. Helper T cells actually will turn on the entire immune system. These control alerting both the B lymphocytes in their production of antibody and the T cells to start engaging in the cell-mediated immunity. So these are very important. These are the functions of the 4 types of T cells categorized by functioning. When T cells are produced, there are actually 2 different types, CD4 cells and CD8 cells. These are those naive cells that have not yet encountered any antigens. Once the T cells have encountered an antigen, then by function we classify the T cells according to these 4 types. Now we're on blood, chapter 17. List the functions of blood. This is right at the beginning of the PowerPoint of chapter 17. Transportation, protection, etc. So you will find these in the PowerPoint script. And then it says list the formed elements of blood, their origins, and the functions of each. Remember, all formed elements of blood, including 1, red blood cells or erythrocytes, 2, all of the various types of white blood cells or leukocytes, and the thrombocytes or platelets are all formed in the red bone marrow. And the PowerPoint script went through the basic functions of each of these. Erythrocytes or red blood cells are the very first formed element that's discussed in the PowerPoint when it starts discussing the formed elements. And then also, I'll come back to the hemoglobin one, but with number 4, it says list the plasma proteins found in the blood. This is also, of course, in the PowerPoint script, and there's a table that lists the various plasma proteins. There's many different types of proteins. Antibodies are one type. Fibrinogen is another one. But the one type of plasma protein that's not discussed too much in other places is that protein, a very important protein called albumin. And that is actually a protein found in blood that allows an osmotic pressure. That means that the albumin, which stays in the blood, will actually pull the water. It provides a force so that the water will stay in the blood vessels. Otherwise, fluid can seep out into the tissues and cause an increase in tissue fluid. Now remember, tissue fluid that leaks out will typically be returned back to the circulatory system by the lymphatic system. But when there is enough albumin in the blood, you won't have a lot of the, it will help prevent filtration of fluid out into the tissue. Number 3 here is the very beginning of the discussion of red blood cells or erythrocytes. Hemoglobin is that protein within red blood cells, and its major function, primary function, is it binds oxygen, O2, and this will bring, the hemoglobin delivers the oxygen to the cells, the living cells in the body, because our body then can obtain energy through aerobic respiration. And our body cells need oxygen to survive. On a short-term basis, skeletal muscle, for example, and other muscle can get some ATP anaerobically, but it's very short-term and it leads to the buildup of lactic acid. So we need to have oxygen brought to the tissues in order to break down glucose to get energy. Hemoglobin is that it has the 4 polypeptide chains with the heme pigment in the center, and that binds an iron atom. And so that is hemoglobin, and red blood cells are packed with hemoglobin. And then 5 is actually still talking about the red blood cells or erythrocytes, and it asks you to describe how the body handles old, worn out, or damaged erythrocytes. And this is on the figure that actually shows the life cycle of a red blood cell. Red blood cells circulate in the body between 90 to 120 days, and they actually have to be broken down and dealt with in a very specific way. So there's that figure in chapter 17, and it shows with red blood cells. Remember, the production of red blood cells is stimulated by erythropoietin, and that's that hormone produced by the kidneys, some tissue in the kidneys that will stimulate the bone marrow to produce more red blood cells when there's a need for them. And as the red blood cells age, they're going to be broken down, and the pigment, hemoglobin, actually will turn into bilirubin, which needs to be removed from the body. It's a yellowish pigment, and the body will remove it primarily as bile, because the bilirubin will go to the liver, and the liver will break it down or turn it into bile, or will conjugate it or turn it into a substance that's water soluble, so that the body can excrete a little bit of it in the urine. But the majority, of course, as bile, will go into the gastrointestinal tract, and then that's going to be converted to stercobilin, which is a brown pigment that is evident in as the normal color of feces. So that figure is a very important figure for you to understand. When the body is doesn't have enough erythrocytes to transport oxygen to the tissues, this is called anemia, and there's several different types of anemia. But what will happen when the body doesn't have enough erythrocytes to transport oxygen to the tissues? It's going to stimulate the tissues in the kidney that makes erythropoietin, and that's going to then, of course, go to the bone marrow to increase production of red blood cells. If you can't get enough, if the body for some reason can't make enough red blood cells, like if there's not enough iron in the diet, or you're not absorbing enough iron, or for any other number of reasons, if the person loses some blood, has some bleeding, that can lead to anemia. Then we're on to the third formed element. Remember, red blood cells are the first one that's discussed in the PowerPoint. The second formed element would be the white blood cells, and these they're actually discussed, I believe, in a short term in chapter 17. Talks about the 5 main types. There's never let monkeys eat bananas. That little phrase can be used to help you remember what the frequency with which we'll find the particular types of white blood cells: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And the little photomicrographs are provided for you in there. But mainly white blood cells are described in their functioning in chapter 21 on immunity. 7, now we're going to that third major formed element, the platelets. And platelets are essential for stopping the bleeding. Hemostasis is stopping the bleeding. So platelets are actually critical for this process, but there's many other factors that are involved in actually stopping the bleeding and clot formation. There's a diagram, there's two diagrams in your study module for lecture exam 1 that illustrate platelet aggregation is one of them, and the other one is showing an abbreviated model of how all these various clotting factors actually work together to form a cascade of reaction, which results in, and this is the most important part of that, all of these together will result in the conversion of this molecule prothrombin into thrombin. And then thrombin, once that's present, will convert fibrinogen, which is soluble, floating around in the blood, just ready in case it's needed. Thrombin will convert the soluble fibrinogen into an insoluble molecule called fibrin. And just as it sounds kind of like a fiber. It's a fibrous, insoluble protein that will now be at the site where the platelets are aggregating. The fibrin will actually weave itself within among these platelets and will pull the blood vessel closed and stabilize that clot. So that's important to understand how all these factors are required in order to effectively form a clot to stop the bleeding. And then there's number 9 is describing clot retraction in the anti-clotting system. Both 8 and 9 are very well illustrated in that short animation that I put within your study module, the module for lecture exam 1. And it's only a couple of minutes long. You can watch it over and over. And he very clearly describes these processes, starting with platelet aggregation, and he even goes through the process of clot retraction and the anti-clotting system. And so please be sure to watch that so that you can understand all of those processes. The clot needs to retract because it needs to not grow too large, and then we need an anti-clotting system so that the clot does not become too large or pieces don't start breaking off and go downstream into a smaller blood vessel. And the number 10 is asking you to define these terms. Hypoxia, this is low blood oxygen. Anemia, which is a lack of red blood cells. There's not enough red blood cells in order to carry the oxygen in the blood. Thrombocytopenia, this is low platelet count. Thrombocytes are also known as platelets. Hematopoiesis, this is the process of, in general, blood cell formation. The other term that is given in your PowerPoint text would be erythropoiesis, which is strictly red blood formation. And I believe it also talks about thrombopoiesis, and that would be increasing the count or making new platelets. Thromboembolism, this is, of course, a thrombus or a clot that's floating throughout the blood vessels, and it gets stuck in a small vessel and can actually block the blood supply to the tissues that are distal or that are served by that particular blood vessel. So the thromboembolus is the actual clot in there, and thromboembolism is when that thromboembolus gets lodged in a small blood vessel. And that is the end. I will do the same thing for the study guide for the heart and circulatory system. Have a great day. Bye-bye.